Good morning students. Hope you all had a wonderful holiday and is feeling a bit relaxed now. So, the chapter what we are discussing is respiration in plants, isn't it? So, in respiration in plants we have already covered some points. So, in that the first was the difference between aerobic and anaerobic respiration. In this aerobic and anaerobic respiration steps, when you are seeing one step is common, what is that? It is glycolysis, isn't it? So, in the glycolysis at last what is produced? Pyruvate or pyruvic acid is produced. So, this process we have discussed, isn't it? EMP pathway or glycolysis is discussed. In that the end product what I got was pyruvic acid. So, with aerobic respiration this pyruvic acid is next step. What they are going to do is that they from the cytoplasm they will move into the mitochondria. In the mitochondria the first process takes place which is called as oxidative decarboxylization. So, where it is taking place? It is taking place in the matrix of mitochondria, means the fluid filled space of mitochondria, inner space of mitochondria, there only what is taking place? Oxidative decarboxylization is taking place. So, here what is happening, that pyruvate or pyruvic acid is reacting with coenzyme A and NAD, and it is reacting in the presence of two substances. One is the magnesium ion and pyruvate dehydrogenase enzyme, and this pyruvic acid is converted into acetyl coenzyme A. Okay, carbon dioxide is released, NADH2 is formed. So, this is oxidative decarboxylization. So, this step we have discussed, isn't it? So, in the glycolysis I got pyruvic acid. How many pyruvic acid did I get? I get 2 pyruvic acid. So, this 2 pyruvic acid is going into the mitochondrial matrix from the cytoplasm now, and in the mitochondrial matrix you can find this pyruvic acid is undergoing a reaction called as oxidative decarboxylization, in which pyruvic acid is reacting with coenzyme A and NAD in the presence of magnesium ion and pyruvate dehydrogenase to form what? Acetyl coenzyme A. After this it is going to enter into a cycle which is called as Kreb cycle. So, what is the next step what we are going to discuss? We are going to discuss about a Kreb cycle, a very important cycle. Here a series of reaction takes place. One product is converted into another one. Okay. So, Kreb cycle is the next topic what we are going to discuss. Okay. This Kreb cycle can also be called as tricarboxylic acid cycle, Kreb cycle, or TCA. This is the just abbreviation, TCA cycle, or citric acid cycle, because the first product which is formed is citric acid. That is why it can also be called as what? Citric acid cycle. Whatever the question is, whether it is tricarboxylic acid cycle or TCA cycle or Kreb cycle or citric acid cycle, the answer remains one and the same. You have to draw this cycle, right? Now, so in this Kreb cycle what is happening? We are going to see. Okay. So, you see this pyruvate I told you which is formed after glycolysis, it is moving into mitochondria. There oxidative decarboxylization is taking place, and what is formed? Acetyl coenzyme A is formed, isn't it? This acetyl coenzyme A, if you are seeing, it is a 2 carbon compound. How many carbon is there? 2 carbon. This acetyl coenzyme A, after moving into the mitochondria, when it is formed, this acetyl coenzyme A, it is reacting with oxaloacetic acid. So, what is this? This is oxaloacetic acid. So, acetyl coenzyme A reacts with oxaloacetic acid and it gets converted into citric acid. So, the reaction is acetyl coenzyme A, which is a 2 carbon compound, is reacting with oxaloacetic acid, which is a 4 carbon compound. This 4 reacts with 2 and it adds up to 6 carbon compound. So, what is the first product form? It is citric acid. That is why the name of the cycle can also be said as what? The citric acid cycle. Okay. So, the first product which is formed is citric acid. This citric acid is next converted into, you can find alpha ketoglutarate acid. So, what is the next product which is formed? The citric acid is converted into alpha ketoglutarate acid. Now, this is a 5 carbon compound, because citric acid, when it is converted into alpha ketoglutarate acid, what is released? Carbon dioxide is released. So, carbon dioxide is released, 1 carbon is gone, the remaining carbon will be how many? 6. So, it is forming a it's 5. So what will be the thing? It will be forming a 5 carbon compound called as what? Alpha ketoglutaric acid, which is a 5 carbon compound. Now this alpha ketoglutaric acid will undergo again a reaction, and it will be forming what? Succinic acid. So again one carbon will be carbon dioxide. So you know in the respiration carbon dioxide is given out, isn't it? So carbon dioxide will be released and succinic acid will be formed. So it will be reduced again a 4 carbon compound. So citric acid was 6, alpha ketoglutaric acid was 5, succinic acid was 4 carbon compound. This succinic acid is converted into malic acid. It is like an isomer, somewhat similar. The structure will be a bit different, but the formula remains the same. So succinic acid is converted into malic acid. This malic acid is in turn converted into what? Oxaloacetic acid. So you just need to remember the things what they are forming. So citric acid is converted to alpha ketoglutaric acid, alpha ketoglutaric acid is converted into succinic acid, succinic acid is converted into malic acid, the malic acid is converted into oxaloacetic acid, and the cycle continues. This oxaloacetic acid reacts with acetyl coenzyme A to form what? Again the citric acid, and this cycle continues, and this cycle we call it as citric acid cycle or the Kreb cycle. Okay. Now listen carefully. So in this reaction, the whole mitochondrial matrix, actually what is happening is that you can find carbon dioxide is released. Okay. So where and all the carbon dioxide will be released? Here the carbon dioxide is released, here also the carbon dioxide is released, right? So 2 places the carbon dioxide is released. Along with that, you can also find that some products are formed in between. So here the NAD is converted into NADH2. So this is one NADH2 is produced. Like that, here also you can find NAD is converted to NADH2. Again here also one NAD is produced. Then anywhere here also you can find NAD is converted to NADH2. So every steps you have to remember where and all the reaction is taking place. Okay. So how many NADH2 is formed? 3 NADH2 is formed, right? Now other than that, what and all is formed? You can find 1 ATP is formed here, isn't it? So 1 ATP is here, and anything else is formed? No. Now again here you can find FAD is converted to what? FAD. This ADP, sometimes they also used to write it down, GDP is converted to GTP. What is A and what is G? You know, A is adenine and G is guanine. Okay. If it is ADP, adenosine diphosphate. If it is GDP, guanosine diphosphate. Okay. So in some text it would have been given like instead of ADP it would be given GDP. It's correct only, no problem. Okay. And thereafter you can find FAD. Okay. FAD is converted into what? FADH2. So you can find 3 things are produced. One is your FADH2, second is NADH2, and third one is ATP or GTP. Okay. So these are the products which are formed when the cycle is taking place. So how many NADH2 is produced? 1, 2 and 3. How many FADH2 is produced? 1. How many GTP or ATP is produced? 1. Okay. So that is regarding your Kreb cycle, what and all is formed. So I will sum it up again. You can find in the Kreb cycle what and all is produced. One is your NADH2 is formed, 1, 2 and 3. GTP or ATP is formed, okay, and one FADH2 is formed. Thereafter you have to write citric acid is converted to alpha ketoglutaric acid, alpha ketoglutaric acid is converted into succinic acid is converted into malic acid, the malic acid is converted into oxaloacetic acid. Again this oxaloacetic acid reacts with acetyl coenzyme A to form what? Citric acid. Okay. Next we are going to see the full forms of these all things. So we have already learned some of the full forms. You know what is NAD, isn't it? So what is NAD? Nicotinine adenine dinucleotide. P if it is there, phosphate. NADH2 is what? Hydrogen phosphate. Okay, nicotinine adenine dinucleotide hydrogen phosphate, NADPH2, like that if you are writing. Now what is this ATP? You know what is it? Adenosine triphosphate, isn't it? So that is ATP. And what is FADH2 or FAD? Flavin adenine dinucleotide. What is flavin? FAD? Flavin adenine dinucleotide If H is there, again you have to add what? Hydrogen. Okay. So, FADH is flavin adenine hydrogen dinucleotide, right. So, these are the full forms. Now, listen, how much amount of ATP is generated? So, in the glycolysis, we have already discussed how many ATPs were generated, 8 ATPs. Now, we are going to see how many ATPs are generated in the Kreb cycle. Okay. Before that, moving into the Kreb cycle, you would have seen this pyruvic acid is converted to acetyl coenzyme A. So, that process we called it as what we called it as oxidative decarboxylization. So, one pyruvic acid, when you are seeing, it is reacting with NAD and it is producing NADH2. So, how many is produced? One NADH2 is produced, right. By one pyruvic acid is giving one NADH2. How many pyruvic acid was produced in glycolysis? 2 pyruvic acid, right. So, again 2 pyruvic acid will give how many NADH2? It will be giving 2 NADH2. 1 will give 1, 2 will give 2. Okay. So, 2 NADH2 will be formed. We have already learnt NADH2. If you are seeing 1 NADH2 is how many ATP molecules? It is equivalent to 3 ATP molecules, right. So, how many ATP molecules will be there? So, 2 NADH2 is there, isn't it? So, how many ATP will be there? 6 ATP. I will make it clear once more. So, with 1 pyruvic acid, I am getting how many NADH2? I am getting 1 NADH2. With 2 pyruvic acid, how many NADH2 I will get? I will get 2 NADH2, isn't it? So, 1 NADH2, if you are seeing, it is equivalent to how many ATPs? 3 ATPs. So, 2 NADH2 is equivalent to how many ATPs? 3 into 2, that will be equivalent to 6. So, that is 6 ATP molecules. So, oxidative decarboxylization of pyruvic acid results in the formation of 6 ATP molecules. So, in glycolysis I got 8 and in oxidative decarboxylization I got 6. So, altogether now I am having 14 ATPs produced. More how it is formed in the Kreb cycle. Okay. So, in the Kreb cycle you see 2 GTP is formed, isn't it? So, I told you here you can write it as GTP also. So, here how many GTP is formed? 2 GTP. GTP and ATP is having same thing only. Okay. So, 2 how it is formed? Because this is 1 pyruvic acid is giving how many? 1 ATP. But there are how many pyruvic acid? 2 pyruvic acid. So, like this again one Kreb cycle also will be there, isn't it? So, they also will produce 1. So, 1 plus 1 is equal to 2 ATP molecules. Okay. So, that is number 1. How many NADH2 is produced by 1 pyruvic acid? You can find 1 pyruvic acid is producing 1, 2 and 3 NADH2. This is 1 pyruvic acid. So, 2 pyruvic acid will produce how many? 6, isn't it? So, 1, 2, 3. Again one more is producing, one more pyruvic acid is producing 1, 2, 3. So, altogether 6. How many FADH2 is formed? FADH2 is only 1. 1 pyruvic acid is forming 1. So, 2 pyruvic acid how many they will form? They will form 2. Is that clear? So, this is 2, NADH2 is 3, ATP is 2, right, because 1 is NADH2 is 3 plus 3, 6. Okay. So, this is been given over here. So, how many ATP is produced? 2 ATPs. How many NADH2? 1 pyruvic acid 3, 2 pyruvic acid they will produce 6. We know that 1 pyruvic acid is 1 NADH2 is equivalent to 3 ATP. Like that they are producing how many NADH2? 6 NADH2. So, 6 into 3 is equal to 18 ATP molecules will be produced. Now, one more thing what you have to know is that 2 FADH2 is equivalent to 4 ATP, which means 1 FADH2 is equivalent to 2 ATP. How much ATP? 2 ATP. So, 2 FADH2 is equivalent to how many? 2 into 2, that is equal to 4 ATPs. Understood? So, in Kreb cycle I am getting 2 ATP, 6 NADH2 and 2 FADH2. Okay. I have written already. I am getting 6 NADH2, 1, 2, 3 like that. 1 pyruvic acid, again 1 more pyruvic acid, 3 plus 3, 6. 2 ATP or GTP, because here 1, another one will also produce 1. 2 FADH2, here 1, another pyruvic acid will produce 1. Okay. So, like that I am getting 2 GTP, 6 NADPH2 and 2 FADH2. 1 ATP is equivalent to 1 GTP is equal to 1 ATP, so no problem. NADH2 is equal to 3 ATPs, that is why 6 into 3 is equal to 18. 1 FADH2 is equal to 2 ATP, so 2 into 2 is equal to 4. So, when you add it up, how many ATPs will be produced? 24 ATPs will be produced in Kreb cycle. So, as you know what is ATP? Adenosine triphosphate, which is the energy currency of the cell, isn't it? So, in aerobic respiration, number of ATP generated is 38. How it is generated? I told you. In glycolysis I am getting 8, oxidative decarboxylization I am getting 6, and in Krebs cycle the highest 24. Altogether it will sum up to 38 ATP molecules. So, during aerobic respiration the glucose is broken down to release how much amount of ATP? 38 molecules of ATP is produced. Okay. So, with this we are coming to the end of the Krebs cycle. Next we will be discussing about electron transport system. Hope this is clear. Next we are going to discuss about electron transport system and oxidative phosphorylation. This can be called as electron transport system or ETS and is also can be called as oxidative phosphorylation. You know what is phosphorylation, isn't it? So, you have already learnt phosphorylation, isn't it, in photosynthesis. What is phosphorylation? Photophosphorylation you have learned, isn't it? With the help of light, what is generated? ATP is generated. So, that one we call it as what? Photophosphorylation. Here light energy is not used, here electrons are used for generating the energy, that is ATP. That is why we give the term oxidative phosphorylation. So, what is the term used? Oxidative phosphorylation. So, we will be discussing two concepts. One is your electron transport system, this one, and the next one is your oxidative phosphorylation. Now, listen carefully students. For electron transport system, we need to understand that this is taking place in the mitochondria, whereas it is taking place, it is taking place in the mitochondria. So, mitochondria, when you are saying you are having an outer membrane, right? So, thereafter you are having an inner membrane. Yes or no? This inner membrane is having much folds like this, the structure will be of mitochondria. Now, this inner membrane, inner to it, this place what it is matrix. Again neatly I will draw. So, when you are seeing, you are having an outer membrane of mitochondria, right? Thereafter you have an inner membrane of mitochondria. So, this convulsions and all which you see, it is a cristae we call, isn't it? So, thereafter this space we call it as what? The matrix. Thereafter you can find this space between the outer membrane and the inner membrane, there is space, right? So, that space we call it as intermembrane space. What do we call them? We call the space as intermembrane space. Space between the outer membrane and the inner membrane, we call it as what? The intermembrane space. So, where is actually the electron transport system taking place? It is taking place in the inner membrane of the mitochondria. Where it is taking place? It is taking place in the inner membrane of the mitochondria. There only the electron transport system is present. Okay. So, we will discuss how it is taking place. See the diagram, look into the diagram carefully. You can find this is the inner membrane of the mitochondria. So, the mitochondria inner membrane, if you are taking means you are just zooming it and seeing, you can find the membrane will be like this. Okay. So, this is the whole inner membrane of the mitochondria. Thereafter, you can find what? The outer membrane will be there, isn't it? So, in between the outer membrane, between the inner membrane and the outer membrane, this one is called as intermembrane space. And inner to the inner membrane, what will be present? Matrix. Okay. So, that is what it's been drawn. So, this is the inner membrane of the mitochondria. Which one is the inner membrane? This is the inner membrane of the mitochondria. Okay. The whole is the inner membrane of the mitochondria. This is the outer membrane is not there. So, here it is the outer membrane. Thereafter, you can find between this inner membrane and outer membrane, that is intermembrane space. Inner to the inner membrane, what will be present? It is mitochondrial matrix. So, this part is called as what? The mitochondrial matrix. So, this is mitochondrial matrix, inner part, okay, and this one is your inner membrane and this one is your outer membrane, and this is your intermembrane space. Between the two membranes, the space which we call it as what? The intermembrane space. Hope this is clear, ma? I don't, I should not see anyone having doubt. I will repeat again. So, when you are drawing a mitochondria, inner membrane, it will be like this. This is the inner membrane. The whole blue is what? The inner membrane. This is the outer membrane. Okay. In between the inner membrane and outer membrane, this space we call it as intermembrane space. This space we call it as what? The intermembrane space. Inner to the inner membrane, what you are having? You are having the matrix. Is that clear? So, this diagram is fine. Right. Now, you see how the process is going to take place here. Electron transport system is going to take place. In the matrix, you can find NADH2 is present. It's produced, isn't it? So, in the Kreb cycle and all, we have discussed that NADH2 is produced. This NADH2 has to be converted to ATP. I told you 1 NADH2 is equivalent to 3 ATP. Yes or no? 1 NADH2 is equivalent to 3 ATP molecules. The question comes how, isn't it? For that only the explanation is being given here. Okay. So, NADH2 is broken down to NAD. So, when this NADH2 is broken down in electron transport system, this NADH2 when it is breaking down, NADH2 is breaking down, it is being converted into hydrogen. It is releasing what? Hydrogen, electrons, and at last they are producing what? NAD. What they are producing? NAD. So, when NADH2 is broken down, it is splitting up into hydrogen ion, that is protons, electrons, and NAD is formed. Okay. The same thing is represented here. NADH2 when it is broken down, this NAD is releasing hydrogen ions, NAD and electrons. Where is this electrons going? The electrons is entering the inner membrane of the mitochondria. There you can find a set of electron acceptor molecules are present. Who are present? Electron acceptor molecules are present. Okay. So, who is this electron acceptor molecules? If you are seeing, they are, the names are here. FMN is the first electron acceptor molecule, followed by FES. Okay. FES. OH2 is given. It's better that you write it down. Ubiquinone. What is it? It is ubiquinone. Okay. UQH2. Just refer your text. It will be more clear. See that one and write it down. Okay. So, thereafter cytochrome B, FES, ubiquinone, cytochrome, cytochrome C, cytochrome A, cytochrome A3. Okay. So, like that you can find all these names are not that important, but you should know that these are electron acceptor molecules. Okay. So, NADH2 when it is broken down, protons are present in the matrix, that is H plus is released in the matrix, but the electrons are moving into the inner membrane. Who is accepting first? Flavin mononucleotide FMN is accepting. After that it is going to this electrons is getting transferred to FES1, FES2. So, FES is iron sulfide, isn't it? Thereafter it is going to ubiquinone, then cytochrome B, then FES, thereafter again ubiquinone, then cytochrome, cytochrome C, cytochrome A, cytochrome A3. Like that you can find this electrons are moving from one electron acceptor to the next electron acceptor. The light reaction you have learned, isn't it? So, there are electron acceptor molecules, they accept the electrons, thereafter they transfer it to the next, isn't it? So, the same here also it is happening. Now, listen very carefully. In this electron acceptor, when you are looking into these electron acceptors, the first electron acceptor FMN, and this is having at particular places they are forming complexes, means these electron acceptor molecules combine together and they make up some complexes. Okay. So, how many complexes are there when you are seeing this electron transport system? There are 4 complexes. So, where is the first complex? This one. This is the first complex. FMN and FES combined together, we call it as complex 1. What do we call them? Complex 1. Means this set of electron acceptor is forming complex 1. Okay. And this alone, this ubiquinone alone is forming complex 2. Right. Then cytochrome B, and till this ubiquinone, it is all electron acceptors are forming what? Complex 3. And the last one is your complex 4. Okay. So, like this, how many complexes are there? 4 complexes are there. This is one complex, this is second complex, this is third complex, and this is fourth complex. Now, listen very carefully what is happening. When these electrons are moving through this electron acceptors, actually they are moving with very high energy. What these electrons are having? They are having energy. They are energy rich molecules. So, when these electron acceptors are accepting and transferring, actually the energy is also getting transferred. The energy is also getting released. And this energy which is released is utilized. It is being used up in pumping the hydrogen which is present in the matrix to the intermembrane space. Understood? So, in 3 places you can find the hydrogen is being pumped from the matrix to the intermembrane space. What are the 3 places where it is? That is only the complexes. You can find first the energy which is released by the protons, means by the electrons, is used in pumping the proton from this to this, right. So, I will repeat again. So, the first complex is present over here, isn't it? So, when you can find what is happening, these electrons are coming, they are having energy. This energy which is released is helping in pumping the hydrogen from here and it is transferred here. So, the first set of hydrogen has come, right. So, 2 hydrogen atoms are moving here. Now, when this electrons are going like this again high energy is released over this ubiquinon. Okay. So, this energy released by the electron is used in pumping the second set of hydrogen into the matrix. So, here one hydrogen set has come, here another hydrogen set has come. So, this is the second complex, right. So, this one is your third complex. In the third complex also, when it is again moving through the ubiquinon, this electron, so they are releasing energy. That energy is used in pumping hydrogen from here to here. So, how many hydrogen set has come? One set, second set and third set of hydrogen has moved. Now, these electrons will pass like this and at last they will be coming to cytochrome A3. Thereafter, here in the matrix there is what is present? Oxygen is present, oxygen atom is present. So, we cannot, oxygen cannot remain as atom. So, we are writing it as half molecule of oxygen. Okay. So, this half molecule of oxygen is combining with this electrons at last and with hydrogen which is present in the matrix, and they are forming what? They are forming water. So, in the respiration you have learned water is also produced, isn't it? So, the water is also formed. So, how it is formed? The oxygen is reacting with the hydrogen and the electrons and they are forming water. So, water is formed by these electrons which is coming. This electrons is accepted by oxygen and hydrogen. All these are combining together to form what? They are forming water. So, when they are asking you who is the final acceptor of electrons, it is cytochrome A3 if it is inside the membrane. But as such, in the electron transport system, who is the final acceptor of electron? It is oxygen. At last, electron is going to whom? This oxygen. This electrons is reacting with the oxygen and they are combining with the hydrogen to form what? They are forming water. Okay. So, this is called as what? Electron transport system. So, electrons are getting transported from this NADH2. When it is broken down, they are forming NAD. Electrons and hydrogen. Electrons are transported through the electron transport system and at last it is being accepted by the oxygen and they are getting converted into water. When the electrons are coming, they are having high energy which is helping in pumping the hydrogen which are present in the matrix to the intermembrane space. So, now what happens is that the hydrogen concentration here it will be more and here it will be less. Now, automatically what will happen when there is a concentration gradient, you know, hydrogen which is present more here will now go back to the matrix. Okay. So, it is a cycle. They have to come back to the place where they have started from, isn't it? So, when the hydrogen is moving here, you can find after some time they will be coming here. Why it is coming like this? Because here the concentration of hydrogen increases and here it is less. So, when it is less here and when more here, towards the concentration gradient, you can find from high to low the hydrogen will be moving. So, that process is given as what? Oxidative phosphorylation. That is what we are going to discuss now. Listen very carefully, what is oxidative phosphorylation? So, hydrogen, when you are seeing 3 places the hydrogen atoms were released, isn't it? So, hydrogens were released means were pumped from the matrix to the intermembrane space at 3 places, right. Now, this hydrogen has to go back to the matrix, okay. So, the hydrogen has to go back, just like that they cannot go. They need facilitated diffusion. So, what is needed? Carrier proteins. Who are needed? Carrier proteins. So, what are the carrier proteins called? They are the F0 and F1 particles. So, we have learned already this one, isn't it? So, in chemiosmotic hypothesis we have covered. So, these are the carrier proteins. So, F0, F1 particles are called as what? The carrier proteins. So, actually what is happening? Listen. So, this is inner mitochondrial membrane. Inner the blue part which you can see is what? That is called as what? The inner mitochondrial membrane. So, this is inner mitochondrial membrane. In the inner mitochondrial intrinsic, you can find F0 particle is present and the eccentric F1 particle is present over here, and they are attached with the help of what? They are attached with the help of a stalk, okay. So, how the diagram is, you can see. So, this is F0, this is the stalk and this is F1 particle, right. Now, how it is on this side, outer side of the inner membrane, here the hydrogen is more and here the hydrogen is less, isn't it. So, what happens? Hydrogen from here has to come here. Who is taking? You can find this hydrogen is being taken or carried by this F0 and F1 particles. So, hydrogen is moving towards higher, the concentration is high. So, hydrogen is going and binding with the F0 particles with high energy. These hydrogens are having the energy, isn't it? With the energy pump only they have moved there, isn't it? So, they are coming, this hydrogen ions are coming, passing through the F0 particles and they are coming as the F1 particle. From here it is reaching, okay. So, in this you can find there is an enzyme present. F1 particle actually act as what? An enzyme, ATPase. How they are acting? It is acting as ATPase enzyme. So, what they are doing? They are helping in binding of one phosphorus to the ADP. So, one phosphate is binding with ADP and they are forming what? ATP. So, one set of hydrogen is moving, one ATP is produced, okay. Any doubt here? So, how many sets are moving? One set of hydrogen when it is moving through this carrier protein. Here you can find ATP is combining with P to form what? ATP. Like this, one ATP is produced and the hydrogen is coming out, right. Like this, how many hydrogen has moved in? 3 hydrogen has moved in, isn't it? So, this 3 hydrogen in the same way they will be coming out. One hydrogen is producing how many ATP? 1 ATP. Like that, how many hydrogen will be coming out? 3 hydrogen will be coming out. 3 hydrogen will be producing how many ATPs? They will be producing 3 ATPs. So, like that you can find if one NADH2 is broken down, this is called as oxidative phosphorylation. Means with the help of this hydrogen oxidation reaction, you can find ADP is combining with P to form what? ATP. Here light energy is not involved. Here the energy of the protons are used up, isn't it? So, that is why we call it as what? Oxidative phosphorylation. So, when one NADH2 is broken down, how many ATP is produced? 3 ATP. So, 1 NADPH2 is equivalent to 3 ATP. You can understand why? Because 1 NADH2 when it is converted to NAD, it is producing how many hydrogen is pumped into? 3 hydrogen. This 3 hydrogen when it is coming out, how many ATPs are produced? They are producing 3 ATPs. So, like that 1 NADH2 is equivalent to 3 ATP molecules, okay. So, in the same way FAD, when you are seeing FAD, when it is broken down, only at 2 complexes hydrogen will be released. So, how many ATPs will be produced? They will be producing only 2. If it is 3 hydrogen released, 3. If it is 2 hydrogen released, 2. Is that clear? So, this process we call it as what? Oxidative phosphorylation. So, we are writing now 1 NADH2 is equivalent to 2 NADH equal to 6 ATP, because 1 NADH2 is equal to how many ATP? 3 ATP. How this 3 ATP is coming? For that only you are having this explanation. Okay. So, through electron transport system and oxidative phosphorylation, I can understand how many ATP is produced. Clear?